In the first lecture on principles of inheritance and variation, we had discussed the father of genetics, some important terms, and also like how Mendel started with the experiments, why he selected the particular type of plant which that is pea plant, and the traits, the characters that he used to carry out his experiments. Now today we will go to learn about the Mendel's first experiment, that is monohybrid experiment. Mendel's monohybrid experiment. In this experiment, Mendel selected only one character, that is single character was selected for his experiment. Now selecting a character means that out of the 7 characters, Mendel considered only one or focused on one character at a time while doing this experiment. Now he created pure lines. He created pure lines for that particular character. Like he selected the alternate traits for that character, and he created pure lines by letting the tall plant to grow for many generations, eliminating if any dwarf plants appeared in any of the generations, collected all the pure plants. Likewise, he selected only the dwarf plants by eliminating if any tall plant occurred while creating the pure line for dwarf. Thus, he created the pure lines. Then he cross-pollinated the parents. Both the parents, that is tall and dwarf, were cross-pollinated. And then they were allowed to grow. The offsprings were allowed to grow. The offsprings which were produced from these parents were called as F1 generation. F here stands for filial. Next, he self-pollinated the F1 individuals. He let the F1 individuals grow on their own. The second generation which was thus obtained, it was called as F2, second filial generation. Now after getting the F2 generation, he collected the plants and then he calculated the total number. There he got the total number. It was actually coming with a ratio of 3:1. We will see further in our discussion how it actually happens. From this experiment, Mendel derived the first and second law of inheritance, which also we will be discussing. Now let us see the experiment in a proper form. Now the parents taken, as we can see here, the parents taken were tall and dwarf. This tall plant was created through the pure line experimentation. This was so for many generations when only tall plants were growing, they were collected, only the tall ones were grown, and after many generations it was ascertained, it was confirmed that they do not have any other gene or any other allele. They have only the factors for tallness. So those plants were selected. Likewise, dwarf plants were selected as the parents. Then from them the F1 individuals were obtained, which came out to be tall. All of them were tall. Not a single dwarf plant was obtained. Then these tall plants were allowed to undergo self-pollination. When they underwent self-pollination, then it was seen that F2 individuals were tall and dwarf. Both tall and dwarf were seen. Now the F1 from these experiments, the conclusions that Mendel derived were that F1 resembled one of the parents. Only one of the parents, that is tall here as we can see, resembled only one of the parents. The trait of the other parent was not seen, that is the dwarfness. The trait dwarfness was not seen in F1 generation. F2 expressed both the parental traits. The next conclusion or inference that Mendel made was that the contrasting characters did not show any blending. The contrasting characters were not showing any blending in the F1 generation. We do not get any intermediate height plant. Then, furthermore, Mendel stated that something was passing down from the parent to offspring through the gametes. Mendel called it as factors. He said that something is passing down. Something is going down from the parent to the offspring through the gametes. That is why we get the traits which were present in the parent, we get in the offspring. If not in the immediate, we get in the next generation. And Mendel called these as factors. Present day, we call them as genes or units of inheritance. Further, some symbols were used to carry out this experiment. Capital letter was used for the trait expressed in F1 generation, like here, and small letter for the other trait. And this experiment, which is with plant height, T for tall. Tall is expressed in F1, so we use capital T, and small t for dwarf, which was not expressed in F1 generation. Now we will see the experiment, how it was actually conducted. Now the parents, tall and dwarf, they were taken. The tall, we use the symbol capital T, and in the parent they will have two of these. It will always be present as a pair. So capital T, and in the dwarf, small t, small t. That is the symbol we are using. During gamete formation, the alleles, okay, they go to form the gametes here. Here also they form the gametes. The F1 generation is then obtained. F1, which comes out to be tall, and this offspring which is appearing in F1 will obviously be having one gamete from one parent and the other gamete from the other parent. Means the genotype, the genetic combination in the plant will be different. That is, they are not similar, unlike the parents. They will be, one will be the trait for tallness and the other will be the trait for dwarfness, but the expression was tall. Then the gametes were again seen. Okay, the gametes that we will obtain, that is, as they are different factors, so we get capital T, small t. The factors, there are different factors present in the F1 plants, so accordingly the gametes will also be of two types. These gametes, they were allowed to fuse. Of course, this was a self-pollinating plant, pea plant, so they were allowed to undergo self-pollination. So upon selfing, we get the F2 individuals. Now to carry out this type of cross, we use this type of grid structure, which is known as Punnett square. Now on selfing, to carry out the cross, the gametes of, say, the male gametes, they are placed on one side horizontally, and the female gametes, they are placed vertically here. So capital T, which is representing the male gametes, say the pollen grains, that will be inside the pollen grains, and the egg cells, that is as female gametes. Now randomly they will meet together. If the factor for tallness in the male gamete and the factor for tallness is in the female gamete, they meet together, it forms a trait that is homozygous or similar factors. Two cases we get dissimilar factors, and again in the fourth case we get similar factors, but this time the similar factors are with a trait or with a factor for dwarfness. So ultimately, when we calculate out of 4 possible offsprings, we will get that 3 are tall and 1 dwarf, which is represented in the form of the ratio 3:1. Of course, the genotypic ratio will be different. That is, here the one tall we get as pure, whereas the other 2 talls, they are quite similar to the F1 generation plants in the having different types of factors. Now, after carrying out the experiment, completing the experiment, Mendel derived the laws of inheritance. The first law, which is also known as law of dominance, it states that Mendel stated that characters are controlled by discrete units called factors. Factors will always occur in pairs in individuals. In a dissimilar pair of factors, one member of the pair dominates, which we will call as dominant, and the other recessive. Like we see in F1 generation, the pair of factors, they are different, capital T, small t. So in such pairs, one of the member will dominate the other. Like say tallness dominates dwarfness. Now the second law states that the law of segregation, it states that factors or alleles of a pair segregate from each other during gamete formation, such that a gamete receives only one of the two factors. They do not show any blending. Now factors or alleles of a pair segregate from each other during gamete formation. When we see the parents, of course it becomes difficult to make out as they are with similar alleles. When we see in the F1 generation gamete formation, during gamete formation it was seen that the two factors, factor for tallness, factor for dwarfness, they segregated, they separated from each other, and the gametes received one each. Some gametes they received the factor for tall, some gametes they received the factor for dwarf. If they would have blended together, then such a segregation would never have occurred, and we can make out that they have segregated when we see the F2 generation offsprings, because we again get different combinations there. We do not always get the same type of alleles there, that is capital T small t, but on the other hand we get all the three possible combinations: capital T expressing tallness, capital T small t, of course tallness but heterozygous form, and small t expressing dwarfness in the homozygous form.